grand tapestry of 20th century science, few threads shine as brilliantly as the contributions of Sidney Brenner. This South African biologist didn't just study life at its most fundamental level. He rewrote the rules for how we understand the very language of existence itself. Born in 1927 in Germiston, South Africa, Brenner grew up in a world where the structure of DNA was still unknown and the mechanisms of heredity remained shrouded in mystery. Yet from an early age, his mind gravitated toward the puzzles of biology. He completed his medical degree and then pursued his doctorate, driven by an insatiable curiosity about how living organisms store and transmit information across generations. The 1960s marked a transformative period in molecular biology, and Brenner stood at the epicenter of this revolution. Working alongside Francis Crick at the University of Cambridge, he helped crack one of nature's most elegant codes. Through meticulous experimentation, Brenner and his colleagues demonstrated how genetic information flows from DNA to RNA to protein. They revealed the existence of messenger RNA, the molecular courier that carries genetic instructions from the nucleus to the protein-making machinery of the cell. But Brenner's vision extended far beyond decoding molecular messages. He recognized that understanding genes in isolation was like studying individual words without comprehending the story they tell. What biology needed was a complete living system simple enough to study in detail, yet complex enough to reveal fundamental truths about development, behavior, and the nervous system. This insight led Brenner to make a decision that would reshape developmental biology. He chose a tiny, transparent worm called Caenorhabditis elegans as his model organism. To many of his contemporaries, this seemed an odd choice. The worm was barely a millimeter long, living in soil and feeding on bacteria. Yet Brenner saw what others missed. This creature possessed exactly 959 cells in its adult form, a number small enough to map completely yet large enough to form a functioning nervous system. Over the following decades, Brenner and his team embarked on an extraordinary journey of biological cartography. They traced the lineage of every single cell in the worm's body, documenting how one fertilized egg divides and differentiates into a complete organism. They mapped the worm's entire nervous system, neuron by neuron, synapse by synapse. This work required patience, precision, and an almost artistic attention to detail, examining thousands of worms under the microscope and reconstructing their development frame by frame. The impact of this research rippled outward in ways even Brenner might not have fully anticipated. C. elegans became one of the most important model organisms in all of biology. Scientists around the world adopted it to study aging, program cell death, gene regulation, and countless other fundamental processes. The worm's transparent body allowed researchers to watch biological processes unfold in real time, like viewing the inner workings of a living clock through a crystal case. In 2002, the scientific community formally recognized Brenner's monumental contributions. He received the Nobel Prize in Physiology or Medicine, sharing the honor with colleagues who had built upon his foundational work. The prize celebrated not just individual discoveries, but an entire approach to biological research, one that emphasized choosing the right organism and asking the right questions. What set Brenner apart was not merely his technical skill, but his philosophical approach to science. He believed in the power of simplicity and finding the minimal system that could illuminate a complex problem. He championed creativity and intellectual risk-taking, often pursuing ideas that seemed unconventional or even impractical to others.